hosted by your Surya Kiran.
आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी
During the COVID, scientists at Sri Chitra Tirunal Institute for Medical Sciences and Technology, that is SCTIMST, have designed and developed a highly efficient super absorbent material for liquid respiratory and other body fluid solidification and disinfection for the safe management and infected respiratory secretions. This product is called as the Chitra Acrylosorb Secretion Solidification System. In this episode, let's meet one of the scientists who contributed towards the development of this product and more. Dr. Manju Saraswati, Scientist E, Sri Chitra Tirunal Institute for Medical Sciences and Technology, Thiruvananthapuram. A brief introduction about herself.
Myself, Dr. Manju Saraswati. I'm a scientist E in the Division of Dental Products, Department of Biomaterial Science and Technology, Biomedical Technology Wing of Sri Chitra Tirunal Institute for Medical Sciences and Technology, Trivandrum. So my background is I'm a MSc in Polymer Chemistry and PhD in Design and Development of Nano Micro Structures Particles for Biomedical Applications. After my PhD, I went to US for my postdoctoral studies, and there means my main concentration is for design and development of polymer formulations for various biomedical applications, including dental, oral drug delivery, cancer theranostics, etc. Means after like 6 years of my postdoctoral studies in US, means I came back and joined at Sri Chitra Tirunal Institute as a scientist D in the Division of Dental Products. So even from the beginning, means I have lot of enthusiasm to see that, means how that particular thing is working, and why can't we have a very effective theranostic or diagnostic devices. So I was very curious to know, means how we can develop, means whether is it possible for us that we can develop that particular things with a little more productive or a little more effective way. So even during my college days, means I have more interest in chemistry and the conjugation things. So when it comes to after my MSc, means I got a chance to work in a project for developing hydrogel. So means that will make and that curiosity, that enthusiasm continues, and I started joined in PhD with Dr. Srinivasan, and he's an academician, and he's having a lot of ideas and a lot of enthusiasm that transferred to all his students, and I also have a lot of ideas and enthusiasm, and during the discussions, and means I got that thing that I have to become a scientist and do more with all these ideas and with things like that. So after my PhD, I got a chance to work in a project based on hydrogels and with Dr. Roy Joseph and Dr. J. Krishnan. And from there itself, I have a lot of curiosity and a lot of enthusiasm to work in this biomedical field. And also, I got a chance to register my PhD under Dr. Srinivasan. He's a great academician, and he is having a lot of ideas, and he passed those ideas to his students and staff. So that way, I also have a lot of passions and ideas, and thinking that I have to work on this, I have to do something more on this particular field, especially for the patient care.
Can you please brief us about your areas of research?
So Currently, I'm more focusing on design and development of materials and devices using polymer formulations for biomedical application, especially dental applications. Some of the project I'm currently doing is developing mucoadhesive bandages for localized oral drug delivery applications and plasticizer-free biomedical devices for medical applications. And also one of the main project that is indigenous product development that I'm doing is developing biodegradable and microparticles for transarterial chemoembolization. And also during the COVID time, we have developed Chitra Acrylosome respiratory secretion solidification system and transferred the technology to Romson Scientific and Surgical Private Limited in Agra. And it's a great product. Actually, this respiratory secretion solidification system is so during the COVID, the main problem is that the contagious respiratory secretions. So the most of the time, the nurses and the nursing assistants who is handling this respiratory secretion, the contagious respiratory secretions, are having facing problems like recontamination. So we have a solution for that particular concept, actually, how we can save the life of nursing and nursing assistant who is handling this contagious respiratory secretion, especially during the COVID. That was the main problem during that time. So what it do is it's based on a superabsorbent polymer system. It's integrated with a disinfectant. So when these respiratory secretions are collected in this secretion canister solidification bag, actually it will be decontaminated immediately because of the presence of a wide spectrum disinfectant, and solidify it because of this superabsorbent mechanism. So it will be decontaminated within a short period of time, and it will be spill free. So and after collecting this secretion, once it is filled, we can seal it because it's a self-sealing mechanism. Cover is there, so we can seal it, and even we can discard it in the regular trash bin because it is decontaminated. So it was it's a great it's very effective during that time, and we did a lot of field trial in our Chitra neurosurgery department for ICU patient, and transfer the technology. After transfer the technology, also we supported the company by doing different field trial in different hospitals, including Anantapuri hospital in Trivandrum and all. So yeah,
That's great. Can you let us know the potential benefits of your research for a common man or the nation's field of science as whole?
So it is highly useful for the common man, actually, because during the COVID it has very high impact because it can decontaminate the secretion within a short period of time. So it's like a decontaminated and spill free. But after the COVID also, we extended the same product for different application, including TB patient and HIV patient, because in all these conditions, because the respiratory secretions are highly contagious, so we can use that canister bag, respiratory secretion canister bag, to collect the respiratory secretions so that it will be decontaminated, and so it will not have there will not be aerosol formation, and it contaminate other patients or other staff in the same. So I'm also working on mucoadhesive bandages for localized oral drug delivery applications. So there are many gel formulations or powder formulations are available in the market for oral drug delivery applications such as oral cancers, oral ulcers, even after surgical perforations, oral candidiasis, etc. But the problem with those gel formulation is that it will wipe out very easily because of the dynamic oral environment while we are eating or while we are drinking, or due to the excess saliva production. All these hinder the adherence of this gel formulation in the mucosal membrane like gum or palate. So that there is a need of frequent application of this gel system, but in most of the cases, elderly patient or patient with severe disease conditions means it's not comfortable for them. So we are trying to develop a muco-adhesive bandage that have very good adhesive strength on the mucosal membrane, especially on the gum and palate, and it will release the drug of application. For example, for the inflammation, it has like steroid and anti-inflammatory drug and antibiotics can be released very slowly for a period of 8 hours to 24 hours. Or for oral cancers means we can have a membrane with anti-cancer drug release for a period of 24 to 48 hours, so that we need to replace the mucoadhesive bandages only twice in a day or something like that. So that will be highly useful. So we have already optimized the formulations, and now we are doing a biocompatibility evaluation of those formulations. So once the biocompatibility evaluation of that bandages is over, we are trying to collaborate with industries like Privas Denpro Private Limited. We already talked to them and they are very interested to collaborate for that particular product. So that is one of the product. And another research and another interesting research that we are doing in our lab is developing a plasticizer free biomedical devices. So in most of the biomedical devices like blood bags, catheters, dialysis tubings and denture adhesives or soft liners means a huge amount of plasticizers are using, and especially the phthalate based plasticizers. So these plasticizers are not chemically conjugated with the base polymer matrix, so it will eventually comes out to the immediate environment. It will diffuse into the immediate environment, so it will affect the macroscopic properties of the system, and also whatever the coming of the plasticizer, that is the phthalate based plasticizers that is coming out from the matrix will cause systemic toxicity.
Thank you, ma'am. I'm sure the listeners had fun learning about devices in biomedical applications. We will talk more on details of competing countries, future scopes of research, and more in the next episode. Until then, dear listeners, do take care and thank you for listening.